Alright, we are going to talk about human genetic disorders. So these are things that happen within the genes of humans that can cause abnormalities in those who are affected by them. So a genetic disorder is an abnormal condition that a person inherits through genes or chromosomes that are caused by either mutations in the DNA of the genes, which is what we've been talking about, or changes in the overall structure or number of chromosomes. Now, when we talked about meiosis, remember we said that it was very important that it divided evenly so that half of the chromosomes ended up in each of the daughter cells. If that doesn't happen, if there's a mistake and one side accidentally gets an extra one, the other side gets less than, or I guess is shorted one, then it can really throw up the number of chromosomes in those egg and sperm cells, which is what causes these changes in overall chromosome number. So first we're going to talk about pedigrees. Pedigrees is a way that scientists use in order to determine what type of disorder it is and how it's being inherited within a family. So it's a diagram or family tree of genetic inheritance, and it's used by geneticists to map genetic traits. It basically helps us figure out what the chances are of offspring having that trait. So you'll notice that it's made up of circles and squares. Circles are girls, boys are squares. Individuals that are fully colored in are individuals with the disorder. So this is a boy with the disorder, this is a girl with the disorder, this is a boy with the disorder, boy with the disorder. Individuals that have half of the circle colored in, that means they're a carrier. Automatically, when you see half of the thing carried in, you know it's a dominant disease, or excuse me, a recessive disease, because if this guy has the dominant trait, that means that they don't have the disease, but they carry the recessive trait that could lead to the disease. Okay, so you'll notice that a carrier is somebody who does not show the disease, but who can pass the disease along. Now notice how these two individuals have a straight line going in between them. That means that they're married. Also notice that from that straight line is a bracket that comes down and down. That means that these are their children. Okay, these are their offspring. And notice they each have a line going to another individual. That means both their children are married. And notice there's the bracket again for both of those for both those offspring, and that means that their children also have children, and they are affected or not affected. So understand the marriage line is a straight line between two boxes, and a vertical line with a bracket on the end is the children of those parents. So we're going to look at this one first. We have an affected individual, not affected, 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 not affected. So that means that we know it's dominant, okay, because this guy has it and two of his kids have it. A lot of people in this diagram have it, so it has to be a dominant trait. Also, understand that we can figure out that if this person was homozygous dominant, that means that all of the children would have it. But since one child doesn't have it, that means this person must be heterozygous. So think about it. This is big H, little h, and this is little h, little h. Okay, that means that these two children inherited the big H from their dad and the little h from their mom, meaning they're still affected. But this guy inherited the little h from dad and the little h from mom, meaning he is unaffected. So that's how you can kind of figure out, it's kind of like putting a puzzle together. Here's another example. So we have dad is affected, mom is not affected. Now here you can figure out that it is a recessive disorder, because if dad has it, right, we can figure out it's going to skip a whole generation. Look, and then it's going to pop back up. And oftentimes recessive disorders skip a whole generation and pop back up. So we can figure out that obviously this person is, let's say, little r. We don't know what this person is, okay, but we know that they have a dominant trait. So that means each of these guys are going to be carriers, little r, so they should be colored in half. They're carriers, but they don't show the disorder. And then in order for their offspring to have it, that must mean that the wife here of this child is also a carrier. So we'll play with this a little bit when we talk about, when we actually try to figure out the genetics of each of these guys. Okay, karyotypes. So karyotypes is literally, they go under a microscope and they can look at your genes, at your chromosomes right before it's getting ready to divide. So here's one karyotype right here. They match them up by length and their banding pattern. So you see that each of these guys have kind of like stripes on them with areas that are darker and areas that are lighter. So we look at that and we can figure out, oh, these are both chromosome 1. These are both chromosome 2, both chromosome 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, all the way up to the X chromosomes. Notice this person has 2 X chromosomes. That must mean this person is female. Okay, you can also see there are 2 copies of each of them, so this would probably be a healthy female according to the karyotype. And same thing here. So notice there is nothing for the Y, so that must mean that there is no boys. These are both girls, and they're both healthy individuals, because if you look, there are 2 copies of each chromosome. So some chromosomal abnormalities. Down syndrome is a big one. That means you have 3 copies of chromosome 21 or 47 total chromosomes. It can result in intellectual disability, heart defects, respiratory infections. Edwards syndrome is a trisomy or 3 copies of chromosome 18. It means you have 47 chromosomes. It's severe intellectual disabilities and malformations in how they look. Turner syndrome is a single X in a female. Remember, females are supposed to have 2 X's, so that means they are shorter chromosomes, so there's only 45 chromosomes. Females only get this. They are infertile, they can't have kids, and they don't have any secondary sex characteristics. So when you go through puberty, all those sex characteristics you notice on the outside, people who have Turner syndrome, or ladies who have Turner syndrome, do not get any of those. Klinefelter syndrome, when there's an extra X in the male, that means they have 47 chromosomes. It happens in males only. They are infertile or sterile. They have small testicles and otherwise normal appearance. You'd never be able to tell, aside from the fact that they will never be able to have children. Okay, so let's look at some karyotypes. So here's one. You'll notice that there are 2 copies of each chromosome until we get to 21. Notice how we have 1, 2, 3. So this is a girl. Here's the X. Notice there's no Y. 2 X's. So this is a girl with 3 copies of chromosome 21, which means this young lady has Down syndrome. So that is what a karyotype is, and that's kind of some different ways we can diagnose genetic disorders. So I hope that made sense. Please write down any questions you have.